Welcome! In this explainer, we're building on our introduction to temporal interference stimulation to really dig into the evidence. We're going to review the data on its clinical efficacy, look at the models that make it all possible, and check out the results from in vivo studies. Okay, so the theory behind temporal interference is pretty elegant, right? But that's not enough. We have to ask the two most critical questions about any new medical technology. Does it actually work? And maybe even more importantly, is it safe? We're going to follow the evidence along the standard scientific path, starting right at the beginning with the preclinical models and then working our way up, step by step, to the data that's relevant for human application. So let's start at the foundation, the animal models. The first question here is a pretty simple one. Does this concept even work in a living biological system? And right away, the preclinical studies gave us a key finding. They showed a direct and pretty significant impact on cognitive function. Specifically, when they targeted the hippocampus with TI stimulation, it actually enhanced associative memory. And this wasn't just something they saw in how the animals behaved. It was backed up by a really robust change at the molecular level. Researchers measured a remarkable increase in a key biomarker for neuroplasticity. That's the brain's incredible ability to form and reorganize its connections. Now, this is where it gets really interesting because it gives us a clear biological mechanism. This 42% jump in BDNF, a protein that helps neurons survive and grow, provides a solid reason for the memory improvements they saw in lab tasks like contextual fear conditioning. And this just perfectly illustrates the two sides of efficacy. On the left, you're looking at direct structural evidence. Those micrographs show an increased density of dendritic spines, literally the physical spots where synapses happen. Then on the right, you've got the functional proof. Calcium imaging shows that TI stimulation is actively recruiting neurons and enhancing their firing during a task.
Okay, so we've seen some compelling evidence that it works, both on a structural and a functional level. But, that's only half the story. Before we can even think about this for humans, we have to establish a really robust safety profile.
The safety data from these early studies is very encouraging. They achieved effective neuronal activation at low current intensities, and crucially, after 12 weeks of chronic stimulation, there was no evidence of tissue damage, and any heating on the scalp was negligible, well within safe thermal limits. All right, so with strong preclinical data on both efficacy and safety, the next logical step is to translate this to humans. But that leap comes with some significant biophysical challenges that we need to carefully model to overcome. And this table really lays out that challenge in black and white. The human skull is orders of magnitude thicker, and the deep brain targets are just so much deeper. Computational models predict you need about 40% higher current intensities to get the same effect at the target in a human. But despite those scaling challenges, the data we have from ex vivo human cortical tissue is incredibly promising. When they applied a similar TI stimulation, these human samples showed a 29% increase in BDNF. This is huge. It confirms that the fundamental neuroplastic mechanism is conserved in our own tissue, with the smaller number likely just reflecting species-specific differences. So based on these studies and the biophysical models, very clear safety parameters have been set up for the first human trials. We're talking strict caps on current intensity and stimulation duration to ensure thermal safety, all while trying to maximize the chance of a therapeutic effect. So we've got a basic profile for safety and efficacy. The next critical challenge is precision. How do you accurately target a deep brain structure like the hippocampus while making sure you don't stimulate the overlying cortex? Well, the answer lies in some seriously sophisticated computational tools. It's called finite element modeling, or FEM. It basically creates a detailed digital twin of an individual's head. This lets researchers work backwards to figure out the perfect electrode placement and current settings to steer the stimulation precisely to the target. And this brings us to one of the most elegant engineering solutions in the entire framework, which allows for real-time, fine-tuned adjustments to where the stimulation is focused. So check this out. As you can see in the diagram, you don't have to physically move the electrodes. By just changing the amplitude ratio of the two input currents, the focal point of that stimulation field can be electronically steered. This is a huge advantage for adapting the stimulation to each person's unique brain anatomy. But there's a fundamental law of physics at play here. There's a trade-off. As this plot shows, the deeper and larger your target is, the more you inevitably increase the electric field in the superficial parts of the brain. And that's exactly why those FEM-optimized electrode setups are
absolutely required to minimize these off-target effects. Okay, now that we've gone over the core science, the safety, and the engineering for precision, we can finally turn to the ultimate question. What specific clinical conditions could this technology potentially treat? For Parkinson's disease, targeting a key motor structure, the subthalamic nucleus, has shown some significant promise. Data from rodent models points to a 65% reduction in akinesia, or that slowness of movement, while early human studies have reported a 40% reduction in tremor. What about epilepsy, especially for the 30% of patients whose seizures just don't respond to medication? TI could offer a non-surgical alternative. And in fact, studies on human hippocampal tissue have already shown that TI can suppress the kind of electrical discharges that lead to a seizure. And for major depressive disorder, preclinical data suggests an ability to modulate key neurotransmitter systems. In rodent models, targeting the dorsal raphe nucleus, a primary source of serotonin, led to a 28% increase in serotonin release right in the hippocampus. The future of this technology is all about pushing that precision even further. We're talking about next-generation hardware, things like flexible, high-density electrode arrays, and real-time integration with fMRI. These advances promise to dramatically cut down on off-target effects, with some proof-of-concept studies showing a reduction from 18% all the way down to just 4%. So, after going through all the evidence, from preclinical studies to translational human data and all the computational modeling, what's the core take-home message here? In summary, the evidence confirms that there's a clear biological effect. TI stimulation induces structural plasticity, and it upregulates key neurotrophic factors. We also have well-defined safety thresholds, but its precision is entirely dictated by the laws of physics and relies on that sophisticated modeling. And this leads to the final point. Its immense clinical potential is highly target-specific, and it depends on advanced hardware and guidance systems to accurately engage those deep brain structures while protecting all the surrounding tissue.